Good evening, everyone, and welcome to the last week lecture for Animal Physiology. I hope all of you have learned a great amount of, you have got great amount of information and knowledge from this particular course, especially from the lecture sessions and from the live discussions. So kudos to you guys that you have persisted in this rigorous course. We have covered a wide variety of animal physiological topics in this particular course, ranging from integumentary system, skeletal system, neural system, and this particular week, especially for week 12. I know that this particular week was pretty heavy and was information overload because we have covered multiple systems in different sessions. But as the overall aim and objective of this animal physiology course is to give you the brief information and knowledge about what comprised of animal physiology. So with that respect, I think that this course justified its name, that now you have the base information about most of the systems which operate in animals, preferably in humans, obviously, as the major focus in this course was particularly with respect to humans only. So I hope that you guys enjoyed learning this course as much as I enjoyed designing these live sessions for you and interacting with you guys and discussing different aspect of things which were not particularly covered in the sessions, but I wanted you guys to start and develop this habit of thinking, right? As we talk and discuss, we start developing to think in particular direction. So that is more important because you can always go back to the lectures. There are multiple resources online available, offline available. You can always learn the basics from these things, but it is important to have a mindset and a vision that you can actually imagine and think and ask questions, irrespective of whether you want to pursue in the field of research or not, right? The important characteristic of any student or a learner or a researcher is the ability to think. So for thinking, you need basic building blocks, basic fundamentals of a system on which you are planning to work, obviously. So this course was designed for that particular purpose only, so that you know what widespread things are available in this arena and what intrigues you, what interests you. So hope this course justified your expectations, and you also learned a great deal of knowledge, and you can say that now you understand animal physiology to some extent. So with this, I hope that all of you have registered for your exam, and all the best for your exam. Don't worry about it. And I guess the pattern of the exam was circulated few days back. So I hope that you guys are clear with that. And don't worry much about the exam. Just follow the sessions, go through the live discussions one more time, go back to your assignment questions which you have solved and submitted. Those things will help you in revising certain topics and also help you in understanding what sort of questions you can expect in the exam. So with this, as always, we're gonna discuss something different in the beginning. It will be related to this particular course, it's this particular week itself, but obviously to great extent we're gonna discuss it, and we'll see that, and then we'll go to our assignment questions, previous year assignment questions, and then we'll wrap up the session. So let me start with discussing about digestive system. So in this particular week, a lot of systems were covered, like endocrine system, blood, digestive system, and other processes also. So I want to focus your attention particularly for the digestive system. Why? Because every single thing, whatever is working, whatever we are doing, we are able to do, and uh, everything else, you know, we are able to do because we have energy, right? And we have already discussed how this energy comes into picture. The food we take, it gets metabolized, the chemical energy is converted into electrical energy, then into molecular energy, ATP, and all those things, right? We have discussed in quite detail in following session, in the like earlier sessions, correct? So every system is important, obviously. Without brain, you cannot function. Without kidney, you cannot function. Without your muscles, you cannot function. So everything at its best or optimal level is what evolution has aimed for, irrespective of which system we are talking about. But why I wanted to discuss digestive system is because digestive system is a very, very extensive system. Correct? It starts from our mouth, correct, and ends at the anus, and then it is also then that particular part is associated with the excretion of the waste material and all that stuff. So why I'm saying the digestive system is important, all other systems are also important, but why digestive system is important? Because this is the direct link between us working properly via utilization of energy, and how we can get the energy is from the digestive system, right? We eat food, the metabolization of the food is done in digestive system, right? Then all other processes happen accordingly after the food is being digested, correct? So that is why I wanted to discuss about digestive system. So I hope that you guys are aware of the digestive system comprises of mouth and oral cavity, then tongue, esophagus or the food pipe as we know about it, then liver, which helps in fat emulsification, uh, liver and gallbladder both. Then we have small intestine. Small intestine has further divisions. Then we have stomach, then duodenum, jejunum, ileum. All these are smaller parts or the major parts of the digestive system. Then on this hand, we have certain glands associated with the digestive system, that is parotid gland, sublingual gland, submandibular gland. These glands are the ones which produce saliva. And our digestion starts with our mouth, right? So there's very small quantity of enzymes which are present in saliva, and our digestion process starts from the mouth itself. Then we have pharynx, stomach, pancreas, spleen, large intestine, then further parts of large intestine such as transverse colon, descending colon, ascending colon, cecum, sigmoid colon, rectum, appendix, anal canal, right? So all these things comprises what? Our digestive system, correct? So in brief, if we want to write about what digestive system is, because this is the pictorial representation of the digestive system, and these are the major parts which digestive system comprised of. So in a subjective way, how can we say, or how can we describe digestive system? We can say the human digestive system, wait, let me get the pointer. Yeah. The human digestive system consists of gastrointestinal tract plus the accessory organs of digestion: the tongue, salivary glands, pancreas, liver, and gallbladder. Digestion involves the breakdown of food into smaller and smaller components until they can be absorbed and assimilated into the body. The process of digestion has three stages: the cephalic phase, the gastric phase, and the intestinal phase. Okay, we're gonna see maybe a little bit about these phases, but overall, as I mentioned, the digestion starts with your mouth, then major digestion part occurs in your stomach, as you guys might be aware of it, and then the absorption part and assimilation part occurs in the intestinal part of the digestive tract. Okay, so to understand this in a better way, I have a brief and very detailed, I personally like this description of the digestive system from this video, and I hope that you guys can also get the same amount of information which I want you guys to have, particularly with respect to the digestive system. Let me share my screen. Yeah, I hope that you guys can see this. Apologies for the ad. I cannot disable them. I'm sorry.
The only phone with best take. Google Pixel 8.
I did. Yeah. So, uh, this digestive system. So there might be different things which you already know about digestive system, and there are things which kind of myth about digestive system. So we might tackle some of these queries during the video as well as while discussing more about the digestive system later. Okay, let me give you that teaching.
The human digestive system is very complex and has evolved over millions of years. It basically consists of the rectum, the large intestine, the small intestine, the pancreas, the stomach, also called gaster and ventriculus, and the liver with the gallbladder. The esophagus is also part of this system, as well as various salivary glands near the mouth. First, the food is broken up in the mouth by the teeth and then mixed with saliva with the help of the salivary glands. Saliva contains a digestive enzyme called amylase that already begins to digest carbohydrates in the mouth. It splits carbohydrates into smaller units. The ball-like mixture of food with saliva, also known as bolus, is pushed into the throat by the tongue, and finally into the esophagus, which propels the bolus to the stomach. The esophageal lumen, that is, the opening inside the esophagus, is very flexible, which allows boluses of different sizes to be transported. The esophagus consists of several layers. These layers occur throughout the entire digestive tract. The two outer muscle layers are responsible for peristalsis. Through these two muscles, the bolus can be transported from the mouth to the stomach, even if the person is standing on his head. The stomach is often divided into 6 areas. The stomach is composed of a similar structure to the esophagus. It has a longitudinal muscle layer on the outside. Underneath, we can find circular muscle fibers. In addition to this, there is an oblique muscle layer overlaying the mucosa. On the inside, there are rugae that allow the stomach to enlarge when food is consumed. The stomach wall contains gastric glands. They produce mucus, which is able to protect the stomach wall from the secreted gastric acid. Gastric acid is produced by simply smelling or seeing food, but also spices, and the stretching of the stomach causes the secretion, that is, the release of gastric acid. About 1 to 2 liters of gastric juice are produced per day. Since the esophagus does not have a protective mucus layer like the stomach and esophagus are separated by a sphincter. It relaxes when a bolus is pushed from the esophagus into the stomach and then contracts to prevent acid and food from going back up. Gastric juice consists, among other things, of hydrochloric acid, the enzyme pepsin, the intrinsic factor, and lipase for the digestion of fat. In addition to nutrients, food also contains bacteria that can damage the body. The components of hydrochloric acid are able to destroy harmful bacteria. In addition, hydrochloric acid converts pepsinogen, also released by the gastric glands, into pepsin. Pepsin is able to break down proteins in the stomach. For a vitamin B12 absorption in the small intestine, the intrinsic factor is needed, which is produced by the gastric glands. The vitamin must combine with intrinsic factor, then it can be absorbed later by the small intestine. Vitamin B12 helps keep the body's nerve and blood cells healthy and helps make DNA. It also contains gastric lipase, an acid-resistant enzyme for fat digestion. In the stomach, gastric lipase splits a triglyceride into a free fatty acid and a diglyceride, whereby only the free fatty acid can be absorbed by the body. More effective fat digestion takes place in the small intestine.
एक बच्चे के हाथ पे जब मैंने खाना का पूरा थाली रखा तो वो बहुत बहुत मतलब
Through gastric juice and stomach movements, which take place approximately every 20 seconds, the individual boluses are mixed to a semi-fluid mass of partly digested food, the so-called chyme. The chyme cannot enter the duodenum at first because there is a sphincter at the stomach exit. The pyloric sphincter resembles the esophageal sphincter. The pyloric sphincter opens only a few millimeters, so that larger pieces remain inside the stomach. In the first section of the small intestine, the duodenum, bile, and pancreatic secretions are mixed with the chyme via the ampulla of Vater. Pancreatic juice contains numerous digestive proenzymes and enzymes. In order for these to do their job, a higher pH value than that in the stomach is necessary. For this reason, pancreatic juice contains sodium hydrogen carbonate. Hydrogen carbonate is able to neutralize the acid in the chyme and thus produce the optimum pH value of 7 or 8. Pancreatic juice also contains proenzymes. It is only through enterokinase, released by the duodenum wall, that the proenzyme trypsinogen becomes trypsin, which can split proteins and activate other trypsinogens. We also find alpha amylase, which we had already found in the mouth. It now does the rest regarding the splitting of carbohydrates, which it converts into maltose and isomaltose. Furthermore, pancreatic lipase is able to split triglycerides into two free fatty acids. The gastric lipase, as we have seen before, can produce only one free fatty acid. The pancreatic lipase can cleave triglycerides excellently because the bile breaks the fats down into tiny droplets. This is called emulsification. Numerous other enzymes are part of pancreatic juice, but these will not be explained in detail here. Bile is produced by the liver cells and transported to the gallbladder. The bile is stored in the gallbladder and finally added to the food in the duodenum via the ampulla of Vater. The pancreas also releases juice via the ampulla of Vater. The small intestine consists of three sections: duodenum, jejunum, and ileum. The ileum continues into the large intestine in the right lower abdomen. The duodenum and the jejunum have circular folds to increase the contact surface with the food. These circular folds extend about 1 centimeter into the lumen of the small intestine. These folds are covered with small finger-like projections called villi. Villi increase the surface considerably. Villi are about 1 millimeter long. Each villus contains blood capillaries and a lymphatic capillary called lacteal, which we will see later. The nutrients, marked green here, are absorbed by the villus and transferred to the blood capillaries.
Hit that pose, take that flick, check your angle, post that. Hit that pose, take that flick, check your.
Some nutrients, such as glucose, do not require a carrier. They are transported freely in the bloodstream. Other nutrients, such as iron, require transport proteins like transferrin. Fats are transported by chylomicrons, which are lipoproteins. The triglycerides to be transported are virtually enclosed in the lipoprotein. Chylomicrons and triglycerides are then transported through the lacteal of the villus. Each villus is covered by even smaller microvilli. They multiply the intestinal surface considerably. The microvilli absorb nutrients and transport them to the inside. The last part of the small intestine is the ileum. It does not have, unlike the duodenum and jejunum, circular folds. The ileum absorbs electrolytes, such as calcium, for building bones, hair, and teeth. Trace elements, such as zinc, for sperm production and the immune system. Vitamins, such as B12, for the formation and maturation of red blood cells. And remaining bile acid, which is transported back to the liver via the bloodstream. As with the esophagus, the food is transported by peristalsis. In contrast, segmentation contractions serve to mix the chyme, which is shown here in yellow and red, to make it easier to see the mixing process. The large intestine is thicker than the small intestine. It is about 1 meter long and surrounds the small intestine. The small intestine is connected to the large intestine via the Bowens valve. It opens when chyme is to pass from the small intestine to the large intestine. The large intestine does not have any villi like the small intestine because most digestible substances have already been absorbed in the small intestine. However, the large intestine has an estimated 100 billion bacteria inside. They are important for many other tasks, such as the production of vitamins and the decomposition of fiber for the body's energy production. Many of these bacteria are an important part of the immune system by killing harmful germs. Through peristalsis, the chyme is transported from the ascending colon to the transverse colon to the descending colon. On its way through the large intestine, water is removed from the chyme. Furthermore, mucus is added for proper excretion of waste. Substances that cannot be absorbed through the small intestine or the large intestine remain in the rectum and are finally excreted through the anus.
I hope now that you guys have a clear understanding, or at least somewhat wide spectrum understanding of digestive system. This was the reason I want to take digestive system for discussion, because it has multiple components associated with the system. So if we talk about, for example, let's say skeletal system, right? So we studied about bones, muscles, and then we went into neuromuscular functioning and all. So anyways, most of the systems are complex, but digestive system is unique in such a way that it has so many parts associated with it, and all of them work separately, right? For example, the only function of mouth is to chew and convert the food into bolus, which we have seen in the video, right? Then that bolus has some other function, because if we are uh, not chewing our food properly, as now you might know, the more you chew your chapati, right? Chapati is what? It's wheat. Wheat is carbohydrate at the end of the day, correct? So the more you chew your chapati, and if you have not noticed it, try it today. The more you chew it, meaning the more you keep the chapati in your mouth, the more sweeter it will taste, right? Why? Because as now we know that the saliva which is being excreted when we are chewing a chapati, which is the major component is carbohydrate, the carbohydrates are getting metabolized via amylase enzyme, right? And carbohydrates are generally sweet, generally, but not all carbs are sweet. Some of them are sweet. So that is why the more you chew your chapati, the more sweeter it will taste, because the carbs in the chapati are getting metabolized via the enzyme amylase. Okay. After this, when you chew, and then the peristalsis happen via esophagus, and then it gets into stomach. Stomach in itself is a very, very fascinating organ, right? So I guess you guys are aware of the pH of stomach, right? It's highly, highly acidic. Imagine HCl is present in our stomach, and HCl is one of the strongest acid which is available out there, and it is literally residing in our stomach. And the moment we even smell something related to food, right, our stomach gets prepared for digesting the food, irrespective of whether the food we have ingested or not, right? So all this such a high acidic pH which prevails in our stomach, how come our stomach is preserved, right? Because the, for example, if I don't know if you guys have seen acid burn victims, right? It's a sad truth, but it is a truth that some people, just for the sake of revenge or something else, they actually throw acids on people, right? So you might have seen such acid burn victims. So the moment your skin touches acid, it literally burns, right? And reason, while performing experiments in the lab or in chemistry lab or in biology lab, all the people who are superior to you, who are guiding you in the experiment, make sure to you that you are handling acids very properly and the way they are supposed to be handled, because they are very corrosive. They will literally burn you if your bare skin touch the acid. So if it is such a strong acid and our system cannot take it, how come our stomach is having that and we don't have any problem with it, right? Even our stomach is made up of cells at the end of the day, like every part of our body. So how come this is happening? The answer is mucus. You might know this. But have you ever thought of mucus is also a biological commodity, right? Because it is getting produced by your stomach for like shielding the stomach cells from the acid. But have you ever thought of how come mucus is not getting um, metabolized or say consumed when such a high content of and quantity also quantity of acid is present? So I'll give this question to you guys to think about, or go and look up the internet. The literature is filled with this. That what mucus is composed of and how come it is resistant to such a high acid content. Even strong metals get digested via certain combination of acids. Then how come mucus is able to protect stomach lining? To protect stomach lining, mucus should be very, very, very resistant to the acid effect, correct? And if something is resistant to the acid effect, which means it is worth studying and worth knowing, right? Because we can use this information to make some translational product which might help certain people, or some in some vivid imagination, whatever you can think of the application of knowing what mucus is made up of, especially for the stomach lining, correct? So it's a very, very open question, which you guys, if you are interested, you can always go back and check. Remaining other part, as you have seen in the video itself, that all the accessory parts such as liver, gallbladder, pancreas, these are the glands which pour their secretion into the intestine and stomach respectively, right? Because stomach is not producing all the enzyme. Have you ever thought why this is the case? So if you have little bit of information about enzymes, right? Most of the enzymes are proteins, correct? And proteins are made up of amino acids, correct? So if you know about protein, you know the three-dimensional structure of a protein is the functional form of the protein. And I am saying most of the enzymes are protein. So for the enzyme to work properly, and here I am talking about digestive enzymes, the enzymes should retain their 3D structure, right? I hope I am clear till here. I'll repeat myself. I am saying most of the digestive enzymes are proteins. And we know that proteins are made up of amino acids. For the protein to function properly, its 3D structure should retain in every stage of the process, in whatever process it is involved in. So in a way, we can say if most of the enzymes are protein, so enzymes should be in their proper 3D structural fold. They should be in their tertiary structure and quaternary structure, whatever structure they are in, functional form, so that they can do their proper function or job, whatever is associated with that particular protein or enzyme here. And which particular enzyme? Digestive enzyme. Correct? If from the basic biochemistry as well as structural biology, we know that for a protein to keep its 3D structure, that is the functional form of the protein, it should be in the optimum environment of temperature, pressure, salinity, and all the factors associated with it. These are some of the major factors which determine the 3D structure of a protein. So if you take a protein and you change the pH in which that protein is present, that protein might lose its functionality, meaning it might lose its 3D structure. Correct? If you change the temperature in which you have kept your protein, it might lose its structure. It not might, it will definitely lose its structure the moment you shift the temperature, which is not the optimal temperature for that particular protein. And every protein has their own range, proper range. It's like a bell-shaped curve, you might have noticed. I'll try to draw it. Apologies for the bad drawing. So if this is the range, and say if this is the pH, let's take the example of pH, and this is the activity. Okay, this is the percentage activity or the total activity. So, and this is, for example, this will be 0, this will be 1, and something like 10, say 10, right? So every protein will have their own different range of functionality, right? So for example, if this is a particular enzyme, or here in this case, a digestive enzyme, this enzyme works maximum or has a maximum activity in this range of pH, right? Correct? Similar graph will be for the temperature. Similar graph will be for the salinity or the salt concentration of the system. Correct. So if you put this particular protein in this pH, it won't work to its maximum. And major reason why it won't work to its maximum is because its 3D structure is lost and it's not able to work properly. So, as I said, stomach is a highly acidic environment, right? So there are certain proteins or enzymes which are optimal in the acidic range. So those enzymes will be present only in the stomach because such conditions are fulfilled in the stomach. While some other enzymes which are not properly functioning at a acidic pH, they won't be present in the stomach because they won't be able to function. Their structure will be lost. They will be denatured. Correct? So such enzymes which do not require such high pH to work properly, those are generally secreted outside the stomach in the duodenum region of the stomach, right? And those enzymes are coming from where? Gallbladder and pancreas. So and the chyme is coming from the stomach. Stomach had a acidic pH. So our system is attuned in such a way that at every stage of the digestion, the environment is prepared in such a way that the maximum activity of particular proteins or enzymes will be achieved. So if we want to digest major part or break down major complex system into smaller ones, the stomach is the place. Now we want to further reduce the size of whatever has been digested, then the duodenum is the place. And then again, the peristalsis and mixing, as we have seen in the video, all those physical factors also indulge in the digestion and mixing of the food. So this is the reason that your stomach is highly acidic and only some enzymes will be present. Also, if you have noticed in this particular video, and I guess from your basic 10th standard biology also, you know that there are enzymes which are present in their inactive form. For example, trypsinogen, pepsinogen, right? These are the enzymes which are present in their inactive form. So they require certain modifications in the surrounding, in that particular protein, so that they can be converted into their functional form and then they will function accordingly. Correct? So why this system is in place? Why you think that enzymes are present in their inactive state? They can be present in their active state, but they're only activated when some stimulus comes. So it is a way of assuring that whatever food is coming, it will be digested only when the food is present. Because our cells are also made up of proteins, lipids, the same commodity which we eat, we are made up of it. Correct? So if there's a constant supply of such enzymes which digest such commodities, chances are it might damage our own system. Correct? So this is like a safety switch that these enzymes will be converted to their functional form only and only when the food is present and the digestion is required. So these are some of the minor details which are associated with, for example, digestive system. A lot can be discussed in digestive system. As I mentioned, it's a very, very, very extensive system, but I will keep my discussions brief with respect to digestive system here. I've given you the overview. I have given you some information which was not present in the video and also in the sessions, and you can explore more with it. If you have any question, feel free to ask or drop in the chat box or wherever you can find me. I'll be happy to answer your questions. So with this, we'll start with our questions for this particular week. So let's begin with it. Yeah. So our first question for today is, erythropoietin is majorly synthesized in kidneys, lungs, heart, or pancreas? Kindly drop your answers in the chat box. You have 1 minute for this particular question, and your 1 minute starts now. Last 10 seconds remaining. I hope that you guys have seen the week 12 lectures. I forgot to ask in the beginning itself, and I hope that you have followed. This was the last session for this particular course. So congratulations again that you have completed this course. Yeah, so the correct answer is kidneys. So if you have followed the session, then this might not come as a shock to you guys. But if you have not, don't worry, we're gonna discuss what erythropoietin is. So erythropoietin, that is EPO in short, is a glycoprotein hormone. So we can divide these two things, right? So what is glycoprotein means? Glycoprotein means it's a protein with carbohydrate associated with it. Glyco means glucose or carbs, right? Hormone, it is a hormone naturally produced by the peritubular cells of the kidney, correct? That stimulates red blood cell production. Renal cortex peritubular cells produce most EPO in the human body. Partial pressure of oxygen directly regulates EPO production. The lower the partial pressure of oxygen, the greater the production of EPO. Erythropoietin stimulating agents are recombinant versions of EPO produced pharmacologically. Examples of erythropoietin stimulating agents are epoetin, derbapoietin, and methoxypolyethylene glycol epoetin beta. ESAs are generally indicated in conditions where there is impaired red blood cell production. So, okay, one caveat about this. Okay, you know that there are many individuals who have climbed Mount Everest, right? And the more higher you go, longitude wise, altitude basically, the partial pressure of oxygen starts decreasing, right? Which means, for example, you are sitting at your place right now, you are very easily, you are able to breathe in, correct? Because the pressure of oxygen in the surrounding is sufficient enough and it's apt for your system to get inside because the pressure is higher outside and less inside. That is why we are able to inhale and exhale, right? We are inhaling and exhaling. But this pressure decreases as we go up altitude wise. Correct? So it is very difficult for us to breathe in and breathe out because we need to put more effort breathing in because the pressure is very less outside, right? So in every single breath, less air will come inside you, and less air means less oxygen. Correct? So those people who plan to climb Mount Everest or any higher trek, they generally go under conditioning exercises. Okay. Or you might have heard about base camps and all those things, right? Because you won't start your trekking, your climbing Mount Everest, for example, today, and within 24 hours you'll reach at the top. No, because your body is not adapted to the changing environment. So there are base camps associated at every checkpoint. The moment a certain uh, a significant altitude increment is observed, okay, so that your body can adapt. Also, before this, conditioning camps are available. So in conditioning camps, what happens is your body is being stimulated. For example, a rigorous exercise routine will be associated, different breathing exercise will be associated, all these things to assure that you can be adjusted to the changing environment. So in these conditioning camps and base camps, what happen is your body is getting adapted. How it is getting adapted? It is getting adapted by changing the rate of production of erythropoietin. Correct? The lesser the partial pressure of oxygen outside, the more erythropoietin you're gonna produce. The more erythropoietin you're gonna produce, the more red blood cells you're gonna have. Does this make sense to you guys? I hope it is clear. I'll repeat myself. The lesser the partial pressure of oxygen, more erythropoietin is gonna be produced or stimulated. More erythropoietin means more red blood cells. And in the end, more red blood cell means more oxygen carrying capacity in your blood. Correct? Because more red blood cells, more blood, more hemoglobin, more oxygen binding cells present in your blood. Correct? This is one of the reason that if you might have noticed, people from higher altitude, if we talk about in India itself, in Kashmir, if you have seen Kashmiri people, they are generally very, like, their cheeks are very red, they are blush, they have natural blush, right? Because they have inherently higher level of RBCs, right? Because they are also living at a higher altitude in comparison to us. Okay, this is the space filled model of the erythropoietin. So you can easily visualize it, and you can see this is a protein, though it is not in the form of ribbons, which you guys might be okay with seeing now how proteins look. This is in the space filled model representation. So these are the different ways you can represent a 3D structure of a protein. Okay. So moving to the next question. Next question for today is, which of these is not a part of testis? Is it epididymis, tunica vaginalis, ductus deferens, and endometrium? You guys have 1 minute for this question. And your time starts now. Those who know the answer should also know why that is the wrong answer and to what part it belongs to. It's a good exercise to have while attempting some questions. Last 10 seconds. The correct answer is obviously endometrium. So you guys know that endometrium is not the part of testis. So testis is the part of male reproductive system. And these are the general things which are part of testis. As we can see, we have ductus deferens here. We have body of epididymis, we have tail of the epididymis, then we have the internal structures in the testes. These are like generally labeled as lobules. The covering around the testes is called tunica albuginea. We have septas, which are distributing into, which are helping in distributing into making the testes into lobules. We have efferent ductus, then we have head of the epididymis, we have pampiniformis plexus, and then we have testicular arteries for the blood supply. So these are the general things which are associated with the testis. Okay. So moving to the next question. Which of these is not part of female reproductive system? Is it fallopian tube, fimbriae, uterine fungus, and cervix? So you guys have 1 minute for question 3. Kindly drop your answers in the chat box and your time starts now. Last 10 seconds to go. The correct answer is uterine fungus, obviously. So these are the major parts which are associated with the female reproductive system. As you can see here, ovary, urinary bladder, symphysis pubis, mons pubis, clitoris, labia majora, labia minora, urethra, vagina, rectum, cervix, and uterus. So here I have the same link. If you remember, in the initial classes, I have showed you some 3D models of the system, right? So I found that also for female reproductive system. Unfortunately, for the male reproductive system, the link was not opening, so I'm not able to share that. But obviously, you guys can always go and visualize it. Let me share my screen. Okay, here it is. I hope you guys can see it. So this is the female reproductive system. As you can see, this is the transverse section here, correct? So what are the parts? So if we talk about the overview of the female reproductive system, so the female reproductive system is the organ system containing the body parts involved in sexual intercourse, reproduction, and menstruation. The external part of the female reproductive system is the vulva, and the internal parts consist of vagina, cervix, uterus, ovaries, and fallopian tubes. If we see cervix, okay, let's start from the outside. So this is the outermost part, which is the vagina. So the vagina is a muscular canal measuring up to 3.5 inches long. Internally, the upper section connects to the cervix at the lower end of the uterus. The vaginal opening is partially covered by the labia majora. The vagina is a sexual organ, enables passage of the fetus during birth and allows menstrual blood to exit the body, right? Then moving inwards, we have cervix. So about 1 inch in length, the cervix is located in the lower region of the uterus. The narrow muscular canal connects the uterine cavity to the vagina. During pregnancy, the cervix forms a barrier between the uterus and the vagina. At childbirth, the cervix softens and dilates to allow the fetus to pass into the vagina, and obviously the birth takes place. Talking about uterus, so we all know what uterus is about. Uterus is the place where the implantation or the baby will be available if the fertilization takes place, and also this is the place where those blood vessels and muscles tissues are formed, which is shed during the menstruation process. Then inner to the uterus, we have fallopian tubes, and further we have ovaries, in which the germinal cells are present, which are responsible for the fertilization and gamete formation and all those things. Okay, so to give an overview of the female reproductive system, we can see this particular video. I hope it is audible to you guys.
We have taken a look at the reproductive system in males. Now, before moving on to the process of fertilization, we need to understand the place where this process takes place. Yes, I am referring to the female reproductive system. The site of ovum production, fertilization, and the uterus are all a part of the female reproductive system. Let's have a look at this important system in this video. Do you remember where we began studying the male reproductive system? Yes, it was the site of sperm production. Similarly, we will begin learning from the site of production of the egg cell, or simply the ovum. So the journey begins from these oval structures called the ovaries, one ovary on either side. Did you notice that the ovaries are connected to these tube-like structures? But they're not connected directly. If we look at it carefully, we find that there are finger-like projections which connect the two. These finger-like projections are very important for receiving the ovum when released from the ovaries. But do they have a scientific name? Yes, these finger-like projections are called the fimbriae. The fimbriae help to hold the ovum released and also help to sweep it to the tubes present ahead. But why does the ovum require the sweeping activity? Can't it travel on its own? No, it can't. Unlike the motile sperm, the ovum is non-motile. It cannot move until it is pushed or swept by some other cells. Now, these tube-like structures that we are referring to since a long time also have a scientific name. They are called as the fallopian tubes. And what do you think will be their importance? Let me guess. Is it that they connect the ovaries to the uterus? Yes, that is definitely one important function. But more important than this is the fact that the fallopian tubes are the site of fertilization. Yes, it is the fallopian tubes where the sperms are able to fertilize the ovum. And from here, the zygote starts dividing and grows into an embryo. Now, where will this embryo go further? Needless to say, the uterus. We know that the uterus, commonly referred to as the womb, is the site of development of a baby. And when the growth of the baby is completed, it comes out of the mother's body through the vagina. Do we know what the vagina is? It is basically a small passage that opens to the exterior. It is the site where sperms are received in the female's body. On the other hand, the same passage is used for passing the baby out of the body during childbirth. Now that you've learned both the reproductive systems, can you tell me which one is more complex than the other one? Just think for a moment.
As we have not seen about male reproductive system, we I guess I have a video associated with it, or at least some questions associated with it. So we'll see these things there itself. Yeah. So it was a very brief video about the female reproductive system, and if I'm able to find some more detailed videos or papers associated with the reproductive system, I'll surely add those links into this particular slide, and then I'll upload it to the center. So our next question for today is, which of the following is a germ cell? I guess it's a pretty easy question, but still, go ahead. Is it hepatocytes, neurons, sperm, or cardiomyocytes? So I have to give you guys 1 minute. So do type your answers in the chat box. And your time starts now. Also, I want you guys to think, what is the difference between a germ cell and a somatic cell, because these are the two different types of cells in our body, right? Germ cell and somatic cell. So what are the major differences between a germ cell and a somatic cell, and why the correct answer is correct and why the wrong answers here in this particular question are wrong? Okay. Last 10 seconds to go. The correct answer is sperm. Obviously, the germ cells are sperm and ova, respectively, in male and female. We gonna see in detail, not too much detail, but yeah, it was quite good information which is available in this. Let me skip the ads for you guys.
Right now, thousands of photos of coral reefs are being uploaded to the cloud.
Our body is made up of cells. These cells can be somatic or germinal. All these cells contain nucleus, which means that they contain chromosomes. The sole cells without nucleus in our body are the red blood cells, which means that they don't have chromosomes. Somatic cells, like muscle cells, intestinal cells, nerve cells, liver cells, white blood cells, and so on. All these cells multiply by mitosis, except nerve cells. The second type of cells is the germ cells, or sex cells. They undergo mitosis to multiply, or meiosis to give birth to gametes. But what do haploid and diploid terms mean? These terms are used to indicate the number of sets of chromosomes in a cell. A haploid cell is a cell that contains N chromosomes in its nucleus, while a diploid cell is a cell that contains two N chromosomes in its nucleus. All the cells of our body are diploid except the gametes, which are haploid.
So, oh, sorry, I didn't realize that I was on mute. So the question says the zona pellucida is present on blastocyst, oocytes, ovarian follicles, or sperm. So you have 1 minute for this particular question. Kindly drop your answers in the chat box, and your time has started, but okay, it's started again. Last 10 seconds to go. The correct answer is oocytes, and obviously this is the basic structure of a oocyte or a egg or a ova, right? So the outermost layer is the corona radiata, then inner to it are the follicle cells. Then we have our main cell, which has cytoplasm, nucleus, and all those basic things. The follicle cells and the plasma membrane, there is a layer in between which is called the zona pellucida. Okay, so I particularly like this video because it has a detailed view of oocyte. So I hope you guys will also enjoy it and will have better understanding of uh things. Okay, so let me share it again. Yeah.
Structure of an ovum. Ovum is the mature female gamete which develops from the primitive germ cells embedded in the cortex of the ovaries. Each of the primitive germ cells gives rise to a number of small cells called oogonia through repeated divisions and growth. Oogonia develop into primary oocytes and then into mature ova. Did you know that the ova or eggs of animals can give rise to a complete new individual within a very short period of time of days or weeks? Doesn't this make the ovum a truly remarkable cell in animals? Come, let's learn more about the structure of this remarkable cell. At the end of this module, you will be able to describe the structure of a mature ovum, structure of an ovum. The ova of humans are spherical, about 0.2 millimeters in diameter. They are found enclosed in the follicles of the ovary. Usually, a single ovum is present in a follicle. The structure of an ovum is not very different from other cells, but characteristic names are given to its different parts. For example, the cytoplasm of the ovum is known as the yolk or ooplasm. The nucleus is called the germinal vesicle, and the nucleolus is known as the germinal spot. The thick, transparent envelope covering the ovum is called zona striata or zona pellucida, and the several layers of cells derived from the follicle adhering to the outer surface are called corona radiata. Let's learn more about the different parts of ovum. Yolk or ooplasm. The cytoplasm of an ovum can be differentiated into an outer transparent exoplasm, also called the egg cortex, and an inner opaque endoplasm called ooplasm. The yolk or the ooplasm is composed of the formative yolk and the nutritive yolk. The cytoplasm of the ordinary animal cell is the formative yolk. The nutritive yolk contains numerous rounded granules of fatty and proteinaceous substances embedded in the cytoplasm. The human ovum has very little of this nutritive yolk, which nourishes the embryo very early in its development. So, the human ovum is described as microlecithal, because microlecithal is a term used to describe small eggs with very little yolk. Germinal vesicle. The nucleus is called the germinal vesicle. The germinal vesicle is large and spherical and initially occupies a central position. But as the ovum grows, it becomes eccentric in position. So, the human egg has polarity. The part of the ovum with the nucleus and the polar body is called the animal pole, and the other part is called the vegetal pole. The structure of the nucleus of the ovum is the same as that of an ordinary cell and is enclosed in a nuclear envelope. Within the nucleus is a well-defined nucleolus known as the germinal spot. Coverings of the ovum. A thick covering called the zona striata or zona pellucida covers the ovum for some time after fertilization. This layer may serve as protection during the early stages of segmentation. The corona radiata is composed of 2 or 3 layers of cells derived from the follicles. These cells adhere to the outer surface of the zona pellucida when the ovum is released from the follicle. The cells of the corona radiata are arranged radially around the zona pellucida, and the innermost cells of this layer are columnar in shape.
Correct. I hope now you have a better understanding of the oocyte structure in general, because in the sessions, due to the time constraints, we are not able to uh, go into too details of these things. So I hope that these sessions might help in covering those knowledge gaps. Okay. Moving to our next question for today. Okay. So what do we have here? We have a question asks, for the occurrence of menstruation, progesterone levels blank and estrogen levels blank. So we need to fill in the blanks. Cool. So for the occurrence of menstruation, progesterone levels increase and estrogen levels increase. Or for the occurrence of menstruation, progesterone levels increase and estrogen levels decrease. Or for the occurrence of menstruation, progesterone levels decrease and estrogen levels increase. And last, for the occurrence of menstruation, progesterone levels decrease and estrogen levels decrease. So you have 1 minute for this particular question. Minute for question 6. Kindly drop your answers in the chat box. Your time starts now. Last 10 seconds to go. The correct answer is for the occurrence of menstruation, progesterone levels decrease and estrogen levels also decrease. So what happens is menstruation occurs here. This is the overall distribution of hormones. So LH is luteinizing hormone, FH is follicular stimulating hormone. This is estrogen and this is progesterone. So you can see that menstruation is happening here, right? And during the menstruation, they are asking what happens to the estrogen and progesterone level. So estrogen is this red one and progesterone is this blue one. You can clearly see during the menstruation, both of these enzymes are, both of these levels are low. Hormones are low, right? Not enzymes, hormones. My mistake. So during menstruation, both progesterone level as well as estrogen levels are low, right? To understand this in a better way, the whole menstruation cycle, we will see this brief video about it. Let me skip the ads for you guys and then
bad news for you.
share.
If you're using Microsoft Office applications like Excel, PowerPoint, or Word,
This might seem hard to believe, but right now, 300 million women across the planet are experiencing the same thing, a period. The monthly menstrual cycle that leads to the period is a reality most women on Earth will go through in their lives. But why is this cycle so universal? And what makes it a cycle in the first place? Periods last anywhere between 2 and 7 days, arising once within a 28-day rotation. That whole system occurs on repeat, happening approximately 450 times during a woman's life. Behind the scenes are a series of hormonal controls that fine-tune the body's internal workings to make menstruation start or stop during those 28 days. This inner machinery includes 2 ovaries stocked with thousands of tiny sacs called follicles that each contain 1 oocyte, an unfertilized egg cell. At puberty, ovaries hold over 400,000 egg cells, but release only 1 each month, which results in pregnancy or a period. Here's how the cycle unfolds. Each month, beginning around puberty, the hormone-producing pituitary gland in the brain starts releasing 2 substances into the blood: follicle-stimulating hormone and luteinizing hormone. When they reach the ovaries, they encourage the internal egg cells to grow and mature. The follicles respond by pumping out estrogen. The egg cells grow and estrogen levels peak, inhibiting the production of FSH and telling the pituitary to pump out more LH. That causes only the most mature egg cell from one of the ovaries to burst out of the follicle and through the ovary wall. This is called ovulation, and it usually happens 10 to 16 days before the start of a period. The tiny oocyte moves along the fallopian tube. A pregnancy can only occur if the egg is fertilized by a sperm cell within the next 24 hours. Otherwise, the egg's escapade ends and the window for pregnancy closes for that month. Meanwhile, the now empty follicle begins to release progesterone, another hormone that tells the womb's lining to plump up with blood and nutrients in preparation for a fertilized egg that may embed there and grow. If it doesn't embed, a few days later, the body's progesterone and estrogen levels plummet, meaning the womb stops padding out and starts to degenerate, eventually falling away. Blood and tissue leave the body, forming the period. The womb can take up to a week to clear out its unused contents, after which the cycle begins anew. Soon afterwards, the ovaries begin to secrete estrogen again, and the womb lining thickens, getting ready to accommodate a fertilized egg or be shed. Hormones continually control these activities by circulating in ideal amounts, delivered at just the right time. The cycle keeps on turning, transforming each day and each week into a milestone along its course towards pregnancy or a period. Although this cycle appears to move by clockwork, there's room for variation. Women and their bodies are unique after all. Menstrual cycles occur at different times in the month, ovulation comes at various points in the cycle, and some periods last longer than others. Menstruation even begins and ends at different times in life for different women, too. In other words, variations between periods are normal. Appreciating these differences and learning about this monthly process can empower women, giving them the tools to understand and take charge of their own bodies. That way, they're able to factor this small cycle into a much larger cycle of life.
It was an intriguing perspective indeed, right? So I hope that now you guys are clear about the menstruation, the cycle, the phases, and also the hormone levels during different phases of menstruation. So our next question for today is, I think you guys might be able to answer this because we have just seen a detailed information about it as well as the graph representing different phases of menstruation. So the question asks, progesterone levels reach its maximum blank and estrogen levels reach its maximum blank. So you need to fill in the blanks, right? So progesterone levels reach its maximum before ovulation, and estrogen levels reach its maximum before ovulation, both before ovulation. Or progesterone levels reach its maximum before ovulation and estrogen levels reach its maximum after ovulation. Or is it progesterone level reach its maximum after ovulation and estrogen levels reach its maximum before ovulation? Last, progesterone levels reach its maximum after ovulation and estrogen level reach its maximum after ovulation. So you guys have 1 minute for this particular question. 1 minute for question 7, and your time starts now. Last 10 seconds to go. The correct answer is after ovulation and before ovulation. It was an obvious question because we have just seen in the previous question itself that what is the graph looks like. So the question was about progesterone levels reach its maximum. So you can clearly see that this is the progesterone level and it reaches maximum just after the ovulation. Okay. And the estrogen level reaches maximum. We can see here the estrogen is the red one, reaches maximum before ovulation. Okay. So this is the case. I hope it was not a difficult question to answer. Moving to the next question. Next question states, the basic functional unit of kidney is it neurons, nephrons, Bowman's capsule, or glomerulus? Okay. So I know you guys don't need 1 minute for this, but I have to give, so that you guys can also think about other options and where you have studied them and whatever information you can recollect with respect to these options. And your time starts now. Last 10 seconds to go. I hope you guys are aware of all other options as they were discussed in the sessions itself, not necessarily in this particular week session, but throughout the lecture we have discussed actually. Yeah. So the correct answer is nephrons, obviously. So if you look at the basic structure of a kidney, so you can clearly see that the outermost part is called the cortex and the inner regions are called medulla, and they are associated with a renal artery which supplies the blood for the filtration purpose. Then we have hilum, the junction. Then we have vein. We have renal pelvis. We have ureter. We have a renal papilla, renal capsule, renal pyramid, major calyx, and minor calyx. And these calyxes have these functional unit nephron. To understand more about the structure of kidney, I have this representative video associated with the structure of kidney, which will give you in-depth information knowledge about the uh, kidney. Let me share it. Sorry.
They only get two pairs. Like, get into
I'm
this
sorry, I
chair.
cannot
Like,
skip
I
this
did this
ad.
with flair. Kick that.
Photo unblur. Engineered by Google. Google Pixel 8.
Now, let's get a closer look. Kidneys are large, bean-shaped organs that are approximately 12 centimeters long, 6 centimeters wide, and 3 centimeters thick in adults. The kidneys perform a lot of different functions, such as the excretion and removal of metabolic waste and foreign substances through urine. The kidneys also activate vitamin D when needed and help maintain the balance of fluid volume, pH, blood pressure, and electrolytes in the body. The kidneys also secrete important hormones, such as erythropoietin, which increases the production of red blood cells. The medial border of each kidney has a concave area called the hilum. This is where the ureter exits the kidney and renal artery, renal vein, and lymph vessels enter and exit the kidney. The functional tissue of the kidney, or parenchyma, has an outer renal cortex and inner renal medulla. The medulla is organized into cone-like structures called renal pyramids and renal columns in between the pyramids, which are extensions of the renal cortex. At the junction between the cortex and the medulla are millions of functional units called nephrons. Each nephron can be divided into its major parts: the renal corpuscle, proximal convoluted tubule, loop of Henle, distal convoluted tubule, and collecting duct. Each nephron starts with a renal corpuscle, which is a spherical structure in the cortex that has a diameter of about 200 micrometers. The corpuscle consists of the glomerulus as well as the surrounding double-layered epithelial capsule called the glomerular or Bowman's capsule. The glomerulus is a bundle or tuft of capillaries that supplies the blood that's filtered to become a fluid called the glomerular filtrate or ultrafiltrate, which typically contains no blood cells or large proteins. The ultrafiltrate initially drains into the capsular or Bowman's space, which is actually the space in between the two layers that make up Bowman's capsule. The outer layer of the capsule is a parietal layer of flat, simple squamous epithelium, and the inner layer is a visceral layer of complex epithelial cells called podocytes. The podocytes have prominent large oval nuclei and cell bodies that wrap around each of the capillaries. The podocytes form narrow slits between the cells that filter the blood to form the ultrafiltrate. The glomerulus also has large cells present called mesangial cells that have irregularly shaped nuclei and can be difficult to distinguish from podocytes in a standard slide stained with hematoxylin and eosin. But they'll often have nuclei that look darker than podocyte nuclei in the image. The mesangial cells have contractile properties to help regulate the amount of blood flowing to the glomerular capillaries. They're also involved in immune defense and repair of the glomerulus. In some images of the renal corpuscle, you'll also be able to see the vascular pole, which is where the afferent and efferent arterioles enter and exit the glomerulus in order to supply and drain the glomerular capillaries. The urinary pole is where the ultrafiltrate is drained from the capsular space into the next part of the nephron, the proximal convoluted tubule. Typically, the proximal convoluted tubule, or PCT for short, can only be seen in cross sections because of the winding or convoluted path it takes through the cortex. This part of the nephron reabsorbs many of the substances the body wants to keep, such as organic nutrients, proteins, as well as most of the water and electrolytes in the ultrafiltrate. The PCT also secretes anions and cations, such as hydrogen and ammonium, into the lumen of the tubule for excretion. The walls of this tubule consist of simple cuboidal or columnar epithelial cells with long microvilli. The cytoplasm in each cell is more eosinophilic than the cells that make up the distal convoluted tubules and collecting ducts, which is why the PCT cells will typically stain a slightly darker pink. Also, their lumen will often form a star-like or stellate appearance. Next is the loop of Henle, which can be split into three parts: the proximal straight tubule, or thick descending limb, the thin descending limb and ascending limbs, and the distal straight tubule, or thick ascending limb. The thick descending limb starts in the renal cortex and moves down into the renal medulla, which is from where this cross-section and longitudinal section of the tubule were taken. The cells of this portion of the loop of Henle have very similar morphology to the previous section of the nephron, with simple cuboidal or columnar epithelial cells that have long microvilli, and a dark pink or eosinophilic cytoplasm. The thin descending and ascending limbs of Henle's loop consist of a simple squamous epithelium and passively reabsorbs water. The wall of the tubule is noticeably thinner compared to the thick descending limb, which is seen in the bottom right of this image. The thick ascending limb starts in the medulla and ascends into the cortex. This tubule is responsible for reabsorbing electrolytes such as sodium, potassium, and chloride. In both this cross-section and longitudinal section of the tubule, we can see that the walls consist of simple cuboidal epithelial cells. These cells commonly have unclear or indistinct borders between the cells. In the longitudinal section, there are neighboring tubules from the thick descending limb that can be differentiated based on the thick ascending limb's more neutral staining, meaning it will have a noticeably lighter stain or color to the cells. At the junction between the thick ascending limb and the distal convoluted tubule is a portion of the tubule with tightly packed cells that face the arterioles entering the glomerulus. This area is called the macula densa. The cells here often have prominent nuclei and taller cell bodies. The cells are able to detect changes in sodium chloride levels within the tubule. This allows them to trigger an autoregulatory response when needed in order to help maintain normal blood pressure and blood volume by increasing or decreasing the amount of water and ions the kidney reabsorbs. Similar to the proximal convoluted tubule, the distal convoluted tubule typically can only be seen as cross-sections because of the convoluted path through the cortex. The main differentiating features of the distal convoluted tubule include its lighter stain and shorter microvilli when compared to the proximal convoluted tubule. This portion of the nephron will reabsorb sodium and chloride, which makes the urine hypotonic. The collecting duct is the final portion of the nephron that drains urine from the nephron into a minor calyx at the apex of a renal pyramid. The walls of the collecting duct consist of simple cuboidal and columnar epithelial cells that have well-defined borders and are also lightly stained. The collecting duct is the last part of the nephron that's able to actively help regulate the body's electrolyte and fluid balance. It does this by reabsorbing water from the filtrate as needed and secreting or absorbing various electrolytes as well. Helping current and future clinicians.
As you can see, it's a very, very complex system of filtering, and how beautifully the arrangement and the cells work out this way, right? So let's move to the next question for today. So our next question is, which of the longest part of, which is the, sorry for the typo, which is the longest part of the sperm? Is it head, neck, tail, or all are of equal size? You have 1 minute. I know you guys don't need 1 minute for this, but still. So 1 minute for question 9, and the time starts now. I guess we can move through this question, right? So obviously the correct answer is tail, as you can see in the representative figure of a sperm. This is the head part, this is the neck part, and this is the tail. And clearly we can see that tail is the longest part. It's a very small video of which tells us about the basic structure of sperm. And yeah, let me share it with you.
My name is Victoria Waskow. I'm an international medical student from Poznań University of Medical Sciences in Poznań, Poland.
Oh, okay. I've removed the brief video. This is quite detailed one. I will suggest you guys, due to the time constraint, I'll suggest you guys to go through it, and it will tell you most of the things which you want to know about the structure of a sperm, as well as the functional aspects of different parts of the sperm. Okay. These links will be available in the slide. Obviously, you know this. So last question for this course and this session is, which of the following statement is true? Is it most sperm cells do not have any centrioles? Centrioles in sperm cells can move across head, neck, and tail. Sperm cells have centrioles in tail. Sperm cells have centrioles in sperm neck. So before attempting this, if we have gone through the previous video, you guys, it will be a pretty easy question for you to answer. But irrespective of that, if you have attended the session, you can easily answer this question. You have 1 minute for this question. And your time starts now. The correct answer is sperm cells have centrioles in the sperm neck. So the brief video which I was talking about is this one. That is why I have skipped that detailed one, which you guys can see on your own. But to get a basic understanding of the structure of a sperm, we can obviously see.
Need a non-collateral education loan for your master's in the United States. Compare 15 banks in just 15 minutes.
It's the same video. Okay, we'll see this video. I guess I've completely removed it.
The human sperm is a motile structure formed of 4 parts: head, neck, middle piece, and a long tail. The sperm head is oval in shape. It looks like an egg, with a narrow cranial part and a wider caudal part. The length of the head is about 5 micrometers. Inside the head, the nucleus occupies most of the space. It contains a haploid number of chromosomes, 23 chromosomes, which is the genetic material of the father required for fertilization. The anterior 2/3 of the nucleus are covered by a cap known as acrosome. This acrosome has an outer membrane, inner membrane, and it contains proteolytic enzymes known as lysosomal enzymes. These enzymes play an important role during fertilization. They digest the structures surrounding the oocytes to enable the sperm penetration of the oocytes. The caudal part of the nucleus contains a small depression called implantation fossa. Caudal to the nucleus, there are 2 centrioles. The proximal centriole is present in the implantation fossa, perpendicular to the axis of the sperm, while the distal centriole is located slightly distal at the neck region, parallel to the sperm axis. Examining a cut section of the centriole, it looks like a cylinder, surrounded by 9 peripheral triplet microtubules. This is called a triplet microtubule because it contains 3 microtubules arranged together. This proximal centriole has an important role after fertilization, as it forms the mitotic spindles required for chromosome separation. The 2 centrioles are surrounded and connected by a banded structure known as connecting piece. The upper part of the connecting piece is covered by a convex cap called capitulum. The convexity of the capitulum articulates with the concavity of the implantation fossa to connect the head with the tail of the sperm. Displacement of the connecting piece from the implantation fossa will result in separation of the head from the tail, forming what is known as decapitated sperm, which cannot swim and consequently cannot reach the oocytes. From the distal centriole, the axoneme will extend till the end of the tail. It forms the backbone of the tail. Examining a cut section of the axoneme, it has a 9+2 structural pattern. As you see, there are 2 central microtubules in the middle, surrounded by 9 peripheral doublet microtubules. And these are called doublet because it is formed of 2 microtubules arranged together. Each doublet microtubule is supported by an outer dense fiber, known as ODF. This forms a complex known as axoneme ODF complex. In the region of the middle piece, The axoneme ODF complex is surrounded by a tightly packed mitochondria in a helical pattern. These mitochondria provide energy required for sperm motility. Regarding the tail, it is formed of two parts, principal piece and end piece. The tail does not have mitochondria. In addition, ODF number 3 and 8 are absent and replaced by two longitudinal pillars of fibrous tissue. Reps of fibrous sheath extend between these two pillars to cover the axoneme ODF complex. And so the axoneme in the principal piece is covered by two fibrous supports, ODF and the fibrous sheath. And finally, in the end piece, the axoneme is directly covered by plasma membrane without ODF and without fibrous sheath.
So as I mentioned, it was a very detailed information about the structure of a sperm, and now you know what are the different parts of sperm as well as what these parts are composed of. Okay, so this is all from my side with respect to this particular course as well as this particular week, that is week 12. So thank you so much for being very, very, very patient with me. And I hope you enjoyed the journey as much as I did with you guys. And you have learned something from me, because I definitely have learned a lot from you guys. So I hope that this course will actually help you in future and in present. Feel free to ask any question you guys have. My website, you can drop a mail to me anytime whenever you want to ask something, whenever you just want to chat about anything. I'm open for it. So for last time, question to ponder upon is Dr. Sujoy Gua was awarded Padma Shri for his scientific contributions. Of many of his contributions, reversible inhibition of sperm under guidance is one of its own kind, one of the very first male contraceptives. Read about the functioning of RISUG. It's a very interesting topic, and I guess that most of you will definitely enjoy it. So just a little bit of information is that though officially today we have completed our course, but I'm planning to have one session next Saturday, same time, where I haven't decided what I'm gonna discuss. So I'm inviting all the suggestions and all the things which you guys might want to discuss or learn about or know more about. So feel free to drop a mail, or even in the YouTube chat comment section, you can drop what you want to discuss. And we'll have one extra session on next Saturday to discuss about anything which you want. Otherwise, I'll plan out something for you guys. Okay. So thank you so much and have a good day. Bye.